Today we are celebrating the birth anniversary of Mahatma Gandhi ji and Lal Bahadur Shastri and the death anniversary of Kamarajar. We salute the leaders. Hello everyone, this is Dr. Sangeeta. Welcome to our channel Zootech. Today we are going to deal a topic on molecular genetics in which we are going to prove that DNA is acting as a genetic material. In today's world, we all know that DNA is acting as a genetic material, but before they did not know which is acting as a genetic material, whether DNA, RNA or the proteins. So various scientists have performed many experiments to prove that DNA was a genetic material. So the first experiment to prove that DNA was acting as a genetic material was given by Griffith experiment. In the year 1928, he was the bacteriologist. He was the bacteriologist to prove that DNA is the genetic material. So, for that he has taken two organisms. Griffith has taken two organisms. One is the Streptococcus pneumoniae. Streptococcus pneumoniae. And the other one he has taken mice. He has taken Streptococcus pneumoniae, the other one is the mice. The Streptococcus pneumoniae is a bacteria and the pneumonia is a fever which is affecting the lungs in the case of mice. So, he wanted to develop a vaccine against this. So, he has taken the Streptococcus pneumoniae bacteria, which is being divided into two types. One is the rough, the other one is the smooth type. One is the rough, the other one is the smooth type. You call it as S type and this one will be the R type. Okay. This is smooth, this is the rough type R and this the smooth is the S type. So rough type, this bacteria, the rough type bacteria, this R type will have no capsules. It will have no capsules and the smooth will have the capsules. So that is the difference between the rough and the smooth. So that is the rough type will have the polysaccharide. The rough will not have the polysaccharide coat and the smooth type will have the polysaccharide coat. So that is the difference between the rough and the smooth type. I hope you understood. So rough type is R and the smooth is the S type. So what he is doing is that, so rough he has taken R type 2. He has taken R2 type. Here he has taken S3 type. Get my point? So S1, S has taken 3. S3 type and the rough is the 2 type he has taken. So now what he is going to do? Now he is going to perform the experiments. So first experiment what he did, the mice has been injected with, yes is what I told you, smooth. When the mice injected with the S type, it died because I told you the smooth, they are pathogenic. They are pathogenic and they are virulent. And this one rough, they are non-pathogenic. They are non-pathogenic and they are non-virulent. Okay, so when the mice is injected with this S type, mice died because this one is smooth, it is pathogenic and virulent, mice died. Second, when the mice injected with the R type, R is what I told you, the rough type. So what happened? Mice survived. It did not affect. So when the mice injected with the rough type, that is R2 type, mice survived. And third experiment he performed, when the mice being injected with heat-killed S-type. Heat-killed S-type. What is heat-killed S-type? Heat-killed S-type is you are going to heat the bacteria, in which the bacteria will all die. When this been injected to the mice survived. Of course, we'll all say, we can all predict, right? So when the heat killed, the bacteria been killed when heating. So of course what happened when injected to the mice? Mice survived. Fourth experiment. This is the most important one. What Griffith did, he combined heat-killed S-type. He combined heat-killed S-type with the rough type. Rough type is what? Live cells. Live. Okay. When injected to the mice, what happened? What happened? Mice died. Now the confusion arose to Griffith. When performing individual experiments with the smooth type, of course it is a pathogenic and virulent, mice died. Rough and treated, when the mice injected with the rough type, mice survived. Heat killed S type, the mice survived. But when he combined both, that is the heat killed S type and the rough type, what happened? The mice died. He couldn't understand why did the mice die. What happened and what was the transforming substance which made the mice die. So, he failed to say about the chemical and the biological processes behind the transforming principle. So, he indicated that some sort of information had been passed from the dead type 3. That is dead type. Yes, dead type is what? S type. Because heat killed bacteria is dead, right? The bacteria have been killed. S type to the live type R. To the live type R, blood sampling showed that the blood of the dead mice contained both live type R cells and the live type S cells. That is both live type, that is S type 3 and R type 2, both were live. Get my point? Both were live. So what happened? The Griffith concluded that R strain bacteria had been transformed by the S strain bacteria. The R strain inherited some transforming principle from the heat killed S bacteria. That is the S strain, heat killed S strain bacteria, which made them virulent. Get my point? So which made them virulent. So he concluded that the R strain bacteria had been transformed by the S strain bacteria. The R strain inherited some transforming principles from the heat killed S strain bacteria, which made them virulent. But what happened? Griffith failed to tell about the chemical and the biological processes behind the transforming principle. So after that, the Griffith's followers, scientists Avery et al., that is Avery, MacLeod and McCarthy, they continued with these experiments and they proved the DNA was the genetic material. This experiment will be uploaded in my next video. Thanks for watching. I hope you all understood.